I'm here. Just trying to make the YouTube stream work.
Mm-hmm. So are you projecting this on a screen? Are people coming in person, or do people watch that on their computers?
People watch this on the computer, and we also stream it on our YouTube channel. People on the internet can see the live stream of it on the YouTube.
Okay. Yeah, I see there are 4 people logged in besides yourself.
Yes. About 50 people signed up for the talk, and I sent them the link for the session.
No, it's working.
I think we can start on time, and people will join us from now on.
Fine.
So welcome everyone. Today we have the honor of having Professor Schultz with us, and he's going to give us a talk about the neural reward mechanism underlying reward learning. And we first have to thanks him for giving us this opportunity. And without further ado, let's have your talk, Professor.
So just to make sure, we have 60 minutes to talk, and then we have time for discussion. Is that?
Yes. It's the standard format that we're following, about 60 minutes talk and then 30 minutes for Q&A, and it's up to you. If you want to have an interactive talk, you can ask questions in the middle, and people can answer in the chat box, and I can give them microphone access and video access if they want to, if they raise their hands.
All right then. Well, thank you very much for the invitation. Good afternoon, everybody. It is 2 o'clock now here in England, and we have a cloudy sky and maybe 15 centigrade temperature, but otherwise a very nice day. I hope you have a good day too. You see in this picture my group in the year about 2017 or 18, and you see a number of extra characters on the top left. This is Bill Stauffer, and you see the reward that we're feeding him when he is performing well. And you see your compatriot, Armin Lack, who's now a professor in Oxford. What I'm talking about today has mostly been done before these people came or with some of these people, but I'm telling a longer history than that. And some of the work has been done afterwards. And I will be talking, as requested, about reinforcement learning and dopamine neurons. And I will, at the end, if you have the time and if you're interested, talk about what we've been doing basically in the last 20 years, which is neuroeconomics, using the dopamine, understanding how the dopamine signal is used for updating decision variables, doing economic decision making. And I try to define all the terms that I'm using, but I'm sorry if I cannot discuss everything in detail. All right, let's go. First, what's a reward? The main idea of a reward is that it keeps the agents, gene carriers, biological organisms alive and ensure propagation of their genes into the next generation. I'm looking at the evolutionary perspective. So the daily function to do that is that the agents need to find substances for survival, like proteins and carbohydrates and fat and vitamins and water, and they have to reproduce, have to find mating partners for gene propagation. And that defines, of course, what a reward is. Rewards are all attractive stimuli, events, objects, situations, and so on that are evolutionarily beneficial. That means the rewards are defined not by the physical or chemical properties. A piece of metal is not a reward, even though it has a physical property. A reward is defined by the usefulness for the survival and the gene propagation of biological agents. So rewards are indeed objects or activities or something, but not just based on their physical properties, but based on their value for survival and gene propagation. That means the rewards are defined by biology and not by physical and chemical properties. Okay, so I'll go give some examples here. So this is a collection of rewards. At the top left in the red frame is food and liquids that contain these substances that we need. And then there's sex as activity that's necessary for propagating genes. These are the primary rewards, basically three rewards: food, drinks, and sex. And then there are additional rewards that help us to use and to find these rewards. Rewards produce addiction. That makes rewards even more attractive, which is maybe a partial evolutionary function. Rewards can be completely misused by drugs. Rewards include risk. That, like in the stock exchange, and risk is a major component of reward. Rewards produce long flights of foraging, in this case over the South Atlantic. This picture about this, these were satellite tracking of the wandering albatross, and this is the 12,000 kilometer flight over the Southern Ocean over several months or weeks that this animal did. And then rewards are discoveries. The Rosetta Stone by Champollion helped to discover understand the Egyptian hieroglyphs. Rewards, and that of course produces knowledge, and knowledge of course is helpful for acquiring the survival necessary substances. And then rewards are pieces of art, like in this case a German expressionist, not very well known, has wonderful pictures, but he died in the First World War at the age of 22 or something. And then at the bottom are the social rewards that myself and a few friends in Los Angeles, after a talk, having sake and beer and sushi. And then rewards are depending on your hormone level, on particular hormones, the Maserati, for example. And then of course you have what's called a common currency. You can evaluate all rewards with one standard reference, and that's money in this case. Okay? And so we can then see how these rewards generate functions in order to acquire them. So one function is learning. You learn things that are good for you. You come back to that. That's called positive reinforcement. You have a higher frequency, probability of behavior towards getting something that is good for you. And you have negative reinforcement. You acquire something else. You go to somewhere else because the thing that you're going afterwards is not good for you. So this is like the fruits on the market in Munich, in Germany. And we don't know what these fruits are by looking at them, but ultimately we learn that some of them are really good and others are not so good. That was the top left. The top right is reward have a separate function that generate approach behavior and drive and support economic choices. And this is a candy dispenser in the close to Death Valley in California, in which the people always went for these little candies. And it took quite a while to make that picture because these candies are very attractive. And there's even a sign you should not put your hand in the candy because this stuff is so attractive. And the bottom is the third reward function. This is emotions. Positive rewards generate positive emotions. They're pleasurable. And when you eat them longer or ingestion longer, you have a state of happiness. It's also called liking. And once you know the rewards, you have a desire. You may want the rewards that serve as a goal or a purpose. At the end, it goes all the way to free will. I mean, the rewards constrain your free will because you're tempted to go to these rewards that are good for you. So in the standard definition of a reward function, people say rewards make you happy. And they say that's emotion, you cannot measure emotions. We take a different approach, in particular when we work with animals, where we cannot measure emotions in a reliable manner, and I cannot communicate them verbally. And so we understand that rewards are anything that produces learning, positive reinforcement learning. And the rewards are anything that you use to choose among that is attractive. So we have a perfectly instrumental definition of rewards that allows us then to use the same definitions for economic choice behavior. And so the economists are basically dealing with the acquisition of rewards with these definitions. Okay, so here's an example of the evolutionary function of rewards. The animals, in this case, a unicellular little cellular organism called paramecium, they replicate and they do well for a while, and you see this curve going up here, and then some species didn't do so well and they disappeared. And this is what I mean. The evolutionary beneficial function of reward is to make sure you don't go into the lower curve, but that you keep reproducing and keep living. This is exactly what a reward is made for. All right, so here is an overview of the brain structures for reward. Basically 4 structures: over the frontal cortex, striatum, dopamine neurons, and amygdala are areas in the brain in which specific neurons are only interested in reward. Not all neurons, mostly the dopamine neurons, most neurons in the dopamine neurons, but in the striatum, over the frontal cortex, and amygdala, this is only a subset of neurons. But when these neurons are coding rewards, they code them without anything else. In the other areas, like dorsolateral prefrontal cortex and parietal association cortex, there are neurons that process reward, but usually together with movement or with sensory stimuli. Even in the cerebellum are neurons that are interested in reward, but always together with movements. So the red marked areas are those that are really have neurons that are really tuned directly to rewards. These are the prime areas, and I'll be talking primarily about dopamine neurons today because I was asked to talk about reinforcement learning, and the dopamine neurons are wonderful neurons that are good candidates for supporting reinforcement learning. Okay, so how do we do this? On the left, you see a dopamine neuron, rather small neuron, and then these neurons generate action potentials, which means the opening of a voltage-sensitive sodium channel in which sodium goes into the cell and potassium out of the cell for a very brief moment, usually less than a millisecond. And we can record these movements of ions as a voltage with electrodes. On the top right shows such a trace. The black trace is a voltage trace. As you can see, 200 microvolts, not a big deal. And then we transform that in TTL signals, and then these through so-called Schmidt trigger, and we feed these TTL signals. This TTL means transistor logic. We feed that into a computer and construct histograms and raster displays at the bottom. Every dot in this bottom is an action potential. The time goes from left to right. We have multiple lines of dots. That's repeated trials, repeated presentation of reward like a drop of liquid. And the top is a histogram that is simply the sum of these dots that are underneath. So this is what I'm going to talk about today as the neuronal signals. Okay, the neuronal signals provide a metric, the rate code, in which the higher rate means a stronger neuronal activity. It could also be the inverse coding, in which a lower rate produces a stronger stimulus. The rate code is actually known for next year, precisely 100 years, by Adrian, who worked in fact in the same building in which I'm sitting right now to give the talk, just one floor higher up. And we will remember this rate code thing. This is very important when we talk later about choices and winner-take-all decision mechanisms. The rate code provides a metric. A higher rate of action potentials, more action potentials per second, means a stronger neuronal response, right? So like 1 meter is more than half a meter. 20 impulses per second is more than 5 or 10 impulses per second. That's the idea. And that rate code is not just invented by the brain, but the rate code comes actually as something that's built in by biology. We see at the left side a hair follicle with a hair sticking out, and at the bottom is a receptor, and that translates the mechanical deflection of that hair into action potentials. And the stronger the deflection, the higher is the rate of action potentials. So from these receptors that are not part of the brain, but they have connected to neurons, from these receptors, the brain has learned the language of neuronal activity. That is, the rate code is the metric of the strength of the neuronal signal, right? And from these receptors, the brain has basically picked up this rate code, and we will see that this rate code also exists with rewards, although the rewards are not entirely defined by a physical stimulus on the receptor. That's the important point. Okay, so let's go. This is an example here, rate code. 0.05 milliliters of juice, like apple juice or something, 0.15 and 0.5 milliliter of apple juice is an increasing amount of reward. The reward is delivered at the vertical line, and we see that the neuronal response is much stronger with stronger reward, with more reward. At the top is a small response, in the middle, decent response, at the bottom, a really nice response. This is what I mean by rate code, more action potentials per time in response to a stimulus. Okay. So let's go through these reward functions. Let's start with learning. Okay, we will come to approach the choice later. We won't talk about emotions. So here is the dopamine signal that we saw. It starts out at the top right. This shows the dopamine neurons in the midbrain. At the bottom, we see our neuron that we have seen. Then we see the amplifier, the electrode with the amplifier. We see the neuronal signal that I just showed you. And then above is the first biological result. We see that the animal sits in a cage in the laboratory with a resting key, with its hand on the resting key. And then it releases this key and moves its hand up into a food box, into which it cannot see but reach. And the right-hand side, we see then the moment of movement onset. That is when the animal releases this resting key. And then the left of that movement onset is the time before the movement, and right to that vertical line is the time during and after the movement. And what we see is that just immediately after the movement onset, there are more action potentials per unit of time. And the top histogram shows then, just on top, the histogram shows that this is actually quite a substantial response. So the idea is that here we observe a neuronal response, an increase in the rate of action potentials when the animal has started moving. So what is this? Is this a movement signal? Obviously it happens during the movement. And now comes the trick in neurophysiology. Of course it's a movement, it's during the movement, but does it really reflect the movement or does it reflect something else? And for that we go to the top graph. And the top graph shows the vertical line is the moment in which the animal then has its hand inside the box and touches this piece of food, which is like a little morsel of apple. It's not a drop of liquid, but it's a morsel of apple. And we see in the top part of this top graph, again, this response, these action potentials that come immediately after touching the food, right? This, you see at the left, the animal, a scheme where the animal's hand goes in the food box and touches this piece of apple that's stuck on a wire. And this is a touch-sensitive wire that produces this line in the center. But again, this is with the movement. Now, is it really movement? And then we go to the lower part of this top graph, and we see when the animal touches the wire without a piece of food being there, there's no response. So the animal has done the same movement. The animal has received very similar sensory stimulation from its hand moving in there, but there's no response. That, of course, is very suspicious. And this is a good example how in neurophysiology you need to select the response or identify the response that is actually by which event it is actually caused. Even though it's in a movement, the response occurs after the movement. It's got nothing to do with the movement. It's only got to do with the touch of the food, of the reward. So it's a very naive approach in which we can understand what's happening in this neuron. Okay, so let's keep going a little bit. What we saw is that the animal touches a piece of food, and we wonder, when we talk of the function of reward and reinforcement, we wonder what that is related to learning. And so we have to define learning. We have to go back to this ridiculous Pavlovian dog in which the animal salivates when it sees a sausage, the so-called unconditioned stimulus. And then you present a bell a little bit before the sausage, and after a while of repeating this, the animal starts salivating to the bell. So the bell has now become a conditioned stimulus that elicits a very similar response as the original reward. And that is the conditioned response, and that is positive reinforcement. The animal licks now more and salivates more to that bell than it had done before, because the bell was associated in a contingent manner with this sausage. Okay? So there is a transfer then of the properties of that reward to the stimulus. That means the properties mean the animal licks and salivates to the sausage. That's the reward on the right-hand side. And after learning, the animal starts to lick to the stimulus. So the behavioral reaction of licking and salivation and so on has been transferred, copied over from the reward to the stimulus. The stimulus is now a reward predicting stimulus, and that's our terminology. The animal sees the stimulus, it predicts the reward, the animal starts salivating in anticipation of that reward. It salivates to the stimulus that predicts the reward, even though the stimulus is not a reward itself. We cannot consume the stimulus. Okay, that's the important point. This blue arrow showing the copying from the reward to the stimulus. Okay, let's see if we see that in neurons. And here we go. Now I was supposed to show these slides separately, so we have to go to that all at once. At the top left, you see the neuronal response to a drop of liquid, in this case, without any stimulus. And a very nice response repeatedly. In the center at the left, you see that now a stimulus has appeared and the neuronal response to the stimulus, but no longer to the reward. And so it looks like the green arrow shows that the neuronal response has been transferred from the reward to this reward predicting stimulus, just like the graph on the top right, the scheme. Okay? All right. So this is very funny that in the center, the neuron no longer responds to reward. I'm trying to tell you that dopamine neurons are coding reward, the signaling reward. And you have a wonderful example where the neuron doesn't bother to signal reward, right? It says reward in the center, in this middle diagram, and there's simply no response. And that's very suspicious. That doesn't make any sense, okay? Until we go to the bottom. And what happens here at the bottom was that we have tested an animal repeatedly, and the animal always moved to the stimulus, and then we were telling jokes in the laboratory. In this case with Thomas Jungberg, a postdoc, many years ago. And the animal realized that we were not paying attention, and the animal did not respond anymore. It did not do a movement. The stimulus came, the neuron responded, the animal had no control over its neuron, but then the animal didn't move. And of course, if you don't move, you don't get a reward. And then the neuron shut off. And you see this little sink there at the bottom, where there's absolutely no action potential for half a second or something. And we thought we had lost the neuron, but the neuron came back. And then we tried it again, and in literally every neuron we tried that, we found the same thing. And then we had to make some sense out of it, and that is in this blue circle or ellipse, where we say, well, what we really have seen is the difference between what the animal is getting and what has been predicted. At the top, there was no prediction, and I have to say there was no prediction for the time. Although the animal knows it gets reward in the laboratory, there was no prediction that it would get the reward at that very moment. In the middle, at the left, in the middle, we see there is reward occurring exactly as predicted. There's no difference between the reward and the prediction, and there's no response. At the bottom, we see there is no reward coming because the animal screwed up, but there is a prediction, which is registered by the neuron. So you get reward which is 0 minus a positive prediction. That, of course, comes out as a negative result. That's the neuron is inhibited. So what this does is the so-called reward prediction error. And we go over to this box that says reinforcement teaching signal, and we see this lambda minus v. And that's exactly how Rescorla and Wagner formulated their error-driven reinforcement learning theory, animal learning theory. Lambda's the reward, v is the prediction, and each trial, the lambda minus v is computed and added with a weighting factor called the learning constant alpha to the current prediction to produce the next prediction. And when you have a positive prediction error, the prediction grows. When you have a negative prediction error, the prediction goes down. And the bottom diagram shows this flow chart in which you have a prediction that produces behavior, and that leads to reward, and then you compare the reward with the prediction that generates the error in red, and then that error is used to update the prediction, and then it goes around and around and around. Every time there's a prediction, the reward, there is an error computed, and if the error is different from 0, there will be an update, either an increase or decrease in the prediction. That's what the dopamine neurons do. That's the basic result. That's the reward prediction error. Everything else now from here on is downstream. All right, we can do a learning test, a learning experiment that's a little complicated to look at. The animal has a choice between two stimuli on the computer monitor. One stimulus gives a reward. If the animal touches it, the other gives no reward. And then we can use new stimuli, so-called new problems, in which we give new stimuli that the animal doesn't know, and the animal has to learn by trial and error. And if the animal learns that repeatedly, at the lower left, you see the learning curve goes up. Within a few trials, the animal has learned the new pictures. And the important thing now is on the right-hand side that at the time of the reward, with a familiar picture, the reward is fully predicted and the neuron doesn't care. That's the top part, which says familiar, first 5 trials. And then at the bottom, then the new pictures come in. A new picture has been shown. This is a single neuron. And then we see trial after 5 trials after 5 trials that initially the neuron responds to when the reward does occur, because let's face it, you have 2 possible stimuli. You touch the right stimulus, you get a reward. You touch the wrong stimulus, you get nothing. So the reward probability when a new picture comes, the prediction is 50%. So when you do get a reward, you have a plus 50% reward prediction error. And as you learn the reward more frequently, and your learning curve goes up, you become 70, 80, 90% correct, and the prediction error goes down. This is exactly shown at the bottom right-hand side. This black response after the red line gradually decreases as the animal learns these trials. And after 20 trials, this has almost disappeared. Okay, right. Okay, and then of course for you guys, when you look at reinforcement learning, you immediately go to the Sutton-Bartow temporal difference learning model. And the temporal difference learning model is simply this, the Bellman equation minus the current prediction, which means you have several rewards in a row, rewards or reward predicting stimuli, which is all the same. And the dopamine neurons actually respond to every one of these single rewards. But then they don't respond if the reward or the next stimulus is completely predicted. So when we go from the top, we have the unpredicted rewards as a response. Then we go to the second line, the CS1. We get a response to the stimulus, but no longer to the reward, what I just showed. And then the interesting thing is you have another stimulus, CS2, that predicts CS1, that predicts the reward. Now you get a response to that CS2, but the CS1 is now predicted, and you get no response, and the reward is predicted, and you get no response, right? So this is the idea that, which I'll show at the graph at the bottom, that the reward prediction error is computed of the total sum, the sum of total reward minus the current prediction. This is in fact what dopamine neurons do. They do the temporal difference learning prediction error. Right. Okay, the TD error, as Sutton-Bartow call it. Oh, and the important part of that is that the dopamine reward prediction error response is time sensitive. The TD model, as you remember, temporal difference model, is a time sensitive reinforcement learning model. And if the dopamine neurons comply with that, they should be sensitive to the time. And I've mentioned that before, that the animal knows it gets a reward in the laboratory, so the reward is perfectly predicted. The only thing that's not predicted is when it gets the reward. And that is tested in this, with this neuron here. It's a single neuron over maybe 50, 60 trials, or at least half an hour or so. And we see that at the top, at the habitual interval, after the stimulus goes off, there's no response to the reward. But then when we delay the reward by half a second, we suddenly get a response to the reward. And we get a depression at the time at which reward should have come. So we're getting two responses, right? We're getting a negative temporal prediction error when the reward does not come, and we get a positive temporal prediction error when the reward comes. And the dopamine neurons show exactly the same thing. A depression when the reward is omitted, and a excitation when the reward occurs surprisingly. Okay, and then we go down again, and we repeat that the neuron doesn't respond, and then we praise the reward earlier, and suddenly the neuron realizes, oh, there's a reward. That's very surprising. Although the reward is always predicted, the time is different, and the neuron responds to that timing. But then that cancels the prediction, and there's no more prediction error. Okay, so this is really important. We're having not just a prediction error, but we have a temporal prediction error. And many neuroscientists confuse this completely, don't realize it. And this is the beauty of the temporal difference learning model. It's a model on time. The delta V over delta T is the crucial prediction error, right? Okay. So, oh yeah, and this is the summary. Basically, if we start at the top, this was the Bellman equation. You may be familiar with. Then you come to TD prediction error. That's basically the Bellman equation minus the prediction error, and then that generates learning. The next prediction is the current prediction plus the weighting, the learning constant weighted temporal difference prediction error, and that comes from the Roscola-Wagner learning rule, the lambda minus v, and that comes from the reticulus Pavlov. So we can now know that the Bellman, the temporal difference learning algorithm from the Bellman equation and the dynamic programming actually has its foundation 100 years ago in Pavlov's dog. So everybody laughs about Pavlov, and I have a huge respect for this Pavlov guy. He may not have realized it, what a revolution he created. And the revolution is this, among other things. You have seen 10 years ago, Demis Hassabis and his people had multiple papers, all in Nature and Science and all these fancy journals, in which he showed, in this case, the game of Go, the Asian game of Go, that the TD learning mechanism, with the additional deep learning algorithms, produces learning better than the best world champion at the time, Lee Sedol. Right? So you know this story probably, and it's spectacular, and that has kind of opened up the public interest in this kind of reinforcement learning and artificial intelligence. And all that goes back to Pavlov's dog and the dopamine neurons. Okay? So then a little bit neurobiology. At the top left, we see the midbrain, substantia nigra, SN, and the ventral tegmental area, VTA, and we see this wave of activity. This is supposed to be the dopamine excitation that moves at a speed of 1.5 meters per second in a monkey or a human from the midbrain into the frontal cortex and the putamen and the caudate. And the top right, you see what happens then, like in the caudate, at the synapse, you have the neuron eye, and you have the dendritic tree with its dendrites, and the dendrites are contacted by the dopamine input, but also from the cortical input. And then the idea is that the dopamine input modulates, hopefully increases the connectivity, the transmission between the cortical input A and the striatal neuron I at this area, which is marked with this gray circle. And an input that's not active from a different cortical area, B, will not be modulated. So the dopamine input modulates the existing neurotransmission and increases it with a positive prediction error, decreases with a negative. And this is, of course, it's a speculation, but it's not completely fantasy, because at the bottom we see what happens when you do long-term potentiation. You see at the control, you see that after long-term potentiation, the synaptic response in the striatum neuron is enhanced to cortical stimulation, and you give it dopamine D1 antagonist, and that plasticity is completely gone. After is the curve of after the synaptic trace after is the same as before. Okay, so there is some indication that this is actually not just a reward prediction error hypothesis, but it actually works. The interesting point comes then that it's not just the dopamines receive the reward information, but they are also producing a behavioral response towards the reward. So at the top left, you see the famous self-stimulation box of Olson Milner 70 years ago, in which a rat touches a lever in order to receive an electrical shock to its brain. And many of the so-called self-stimulation sites are linked to dopamine neurons, about half of them, which is shown in the center. Corbett and Wise, 1980, so 45 years ago, have mapped out the effective spots in the midbrain of a rat. These are on the right-hand side, the black dots, and mapped them out and put the map on the left-hand side to make it visible. And we see that only the anterior parts sustain self-stimulation because these anterior parts collect all the axons from the posterior neurons. And then the bottom left shows that with modern optogenetic methods, you get the same result. You optogenetically stimulate the identified dopamine neurons, and you see that the nose poke increases continuously over several days. This is from Ilana Witten in Dijsselrath's laboratory. And then the next slide in the center at the bottom shows that when you actually inhibit dopamine neurons, as Christian Lüscher's laboratory with Tan et al. Has done, you get a reduction in the preference for the chamber. It's a two-chamber choice task, and the animal does not go in that chamber anymore, or much less in that chamber where it gets a dopamine inhibition. So it works in both directions. So it's a bidirectional reinforcement signal that is elicited in this case by artificial dopamine excitation or inhibition, optogenetically defined. Okay, there was heterodopsin in the center. And we can do that in the monkey as well. Bill Stauffer in my lab, one of the people in the picture in the beginning, has stimulated dopamine neurons, both electrically and optogenetically. You see the two curves at the bottom. Electric stimulation is a bit more effective because it directly goes for the voltage-dependent sodium channels, whereas optogenetics only depolarizes the action put the sodium channels, and that then depolarizes the voltage-sensitive sodium channels. And what you see is at the top, the monkey has a choice between the red stimulus and the blue stimulus. When it chooses the blue stimulus, it gets a dopamine excitation through optogenetic stimulation in the midbrain. And when he chooses the red stimulus, he gets nothing. And you can see in this diagram at the bottom that the animal initially has random choice, 0.5 choice probability, and that choice probability rather quickly, after about 20 trials, goes up, and the animal keeps going only for that stimulated option. That means the dopamine signal drives the animal, the agent, towards more reward and away from less reward. That is a very interesting phenomenon that we can exploit. So in this case, we see at the very left, we start out with a prediction. We give the animal a reward that's bigger than the prediction. Then it generates a positive reward prediction error, positive RPE, and the dopamine excitation. That dopamine excitation, of course, increases the prediction through reinforcement learning. And then you need a larger reward, the blue reward, in order to generate the same reward prediction error. And that then generates the same dopamine signal as before. And that keeps going. You take the third graph, and again, the dopamine signal has increased the prediction, you need more reward, and then to get the same reward prediction error, and then you get the dopamine signal, and so forth. So the idea is that by stimulating the dopamine neurons, these animals are actually eliciting an artificial dopamine reward prediction error signal. They're seeking a positive reward prediction error in order to get that positive reward prediction error signal. And the problem is that this positive dopamine reward prediction error signal through reinforcement learning increases the prediction. And with the increased prediction, you need more reward in order to get a positive reward prediction error again. And then the circle continues like this. And this is the whole circle. You seek dopamine excitation on the left, you seek the better reward to predict it to get this dopamine response, you get the reward, and then the excitation, dopamine excitation increases the prediction, you go down, you need a higher prediction, a higher reward, and that turns the circle. And in this case, this iteration going in this recursive mechanism, going through again and again and again, the animal is seeking larger and larger rewards because it wants this dopamine excitation. And it won't get the excitation anymore when the prediction is too high. So it needs an even better reward. So the animal is kind of pressurized to get more reward. This is a mechanism in which the animal is not neglecting the environment, but is really seeking these more rewards. Okay. So this is the interesting thing in reinforcement learning that many people overlook. Reinforcement learning is actually an open-ended process. The error on the left goes to update the prediction produces behavior, the reward, the error comes again. And this prediction can rise, can grow forever until you find no more reward that's better. Then there was no more prediction error, and then it doesn't increase. So you overall increase the reward through this increasing prediction. Whereas the error correction mechanism, which is basically the same thing, but here you have a set point, and the error is then the difference between the set point and the behavior. And what you do is not to increase the set point, but to adjust the behavior so that the error goes down. In both cases, in reinforcement learning and in error correction, the error goes down. But in the case of the error correction, you never go way above what your current set point is. That's the whole idea. You stay at the set point. Whereas the reward prediction error with the reinforcement learning increases the prediction and thus increases the reward. It's an evolutionary, very beneficial mechanism. Okay. So here we go. Why do we go to this pub? Wonderful people, wonderful food, wonderful environment, super atmosphere. We seek excitation of dopamine neurons. This is what our brain does. Our brain doesn't want reward. Our brain wants a dopamine excitation. And with that dopamine excitation, in order to get it, we need to get a reward. And that reward helps us, our body, to live and to reproduce. This is the idea. The dopamine neurons are doing it. They are not doing it alone. There's certainly the striatum and the orbitofrontal cortex and the amygdala, but that's at least one mechanism we can directly link to a neuronal system. Okay? So then the question is, when you start working on the dopamine system, you hear a lot of stories. And one story is that dopamine doesn't do only reward, it does movement, and that is true. Oh, sorry, before I get there, I have to talk about attention. What you realize is that dopamine neurons respond to stimuli that are not rewards. That's very confusing, and you get wonderful comments from Nature editor that there was a discovery 2 years ago about dopamine coding attention. Of course it does, and that's known for 40 years now. And here's an experiment 15 years ago in which I was involved with Masamichi Sakagami in Japan, in which he used a random dot motion pattern with which he was very familiar. And when this pattern came on, then a few of these dots started moving in one coherent direction, and the others went all random. And the animal had to detect whether this coherent motion goes to the right or to the left. And when everything moves very coherently, like 50% of the dots move coherently, it's very easy to see whether they go to the right or the left. And the neurons show two responses at the top. They show a initial sharp response, and then the response goes down, and they show a second response. And the time of the reward, there's no prediction error. But then when you reduce to coherence to 25%, then the thing comes on, but the animal doesn't always get it right. In fact, it gets it 85% correct and 50% wrong. And that means when the animal gets it wrong, the reward value is lower. Less reward probability means less reward value. You still get the first response, that's the first peak in the green zone, but then the peak in the pink zone goes down. That is apparently reflecting the reduced reward. And that is very clear when we go to the bottom, in which there's no coherence, and the animal has to guess. In which case the reward probability is 50%. It's either goes left or goes right. So 50% is correct. He still gets the same initial response in the green area, but the pink response is completely gone. That means the reward, there's no prediction error anymore. The reward is right in the middle of the prediction. And when the animal does get a reward in these 50% of correct trials, you get a huge reward prediction error. So what this shows is what the diagram on the left is trying to say, that the rewards have two components. They have a physical impact. That's you first detect that something happens, and after a while you realize what it is, and you evaluate that reward. And the dopamine neurons do exactly the same thing. They have this initial response that's like, bang, there is something. And then later, the same neurons start analyzing, understanding what the reward value is. And that's why the pink response goes down, because the reward value goes down because of the decreasing probability of getting a reward, right? So we now have two response components in dopamine neurons. Dopamine neurons do two things. Dopamine neurons do attention or stimulus detection, or however you want to call it, and of course our reward prediction error signal, okay? Let's keep going with this idea of multiple dopamine function. When we started out with dopamine neurons, we had a monkey doing a big movement, and we got lots of very heterogeneous activity. 1/3 of the neurons were excited. Some neurons were inhibited during the movement. Some neurons were excited during the reward, and some neurons were excited during the whole task on the left-hand side. But then came Melen DeLong and did a very nice controlled movement task in which there was absolutely no dopamine activity, no response. And then we looked at our data again, and we saw that when we improved the movements, had a much better controlled movements, we still got the response to the stimulus, the phasic response on the right-hand side, but we did not get any movement response. So the neurons were not interested in the movement. And at the bottom, we see eye movements, which are very strong movements, and the dopamine neurons don't care for the eye movements. So the idea is the dopamine neurons are, yes, on the left-hand side, they're involved in movements, they're active in movements, but when you look very close at the movements, there's no activity with the movements. And the result is, the conclusion is that I think you need a lot of movements, in a big movement, to activate the dopamine neurons. And that became very clear with the optogenetics that focused research on rodents. And if you have ever worked with behaving rodents, you realize that it's very difficult for these rodents to sit still. I mean, you can fix the head and you can make them lick right and lick left and do very controlled movements or paw movements. But when you look very closely, you can see that in the movies that are in the supplementary material of the published papers, that the animals move half the body. That means like they're moving 300 muscles. And that's pretty much the same thing in which we saw in our initial less controlled situation. The monkey saw dopamine activity. And sure enough, you see the dopamine activity in these mice when they run down a T-maze. So they're using 600 muscles to run down a T-maze. And they get closer and closer to the goal, and they run faster and faster. And then they turn around, which is a huge movement task. And the neuron activity increases even more all the time. This is actually dopamine release. On the right hand side, running the wheel or trackball. I mean, this animal moves its 600 muscles to stay on top of the little trackball. And of course, you can then correlate the activity with acceleration or speed or movement start, as you have done. So you see the movement activity again. And the resolution came in an experiment by an Anne Churchland 6 years ago, in which she had an animal like that, a rat or a mouse, well trained in the task. And then she very diligently used the multiple linear regression and computed, this is shown in green at the left, the partial regression coefficient, R square, partial R square. And what she saw is some significant R squares in the task. And then with some additional instructions, she also saw some. But then what she saw was even more interesting on the side. On the right-hand side, a lot stronger R squared. That means a stronger correlation with behaviors that were not part of the task. That means what I'm saying, that the animal does the task nicely and correctly, but the animal does a lot of other stuff, like body movement and whisking and nose movement and whatever you want. And so what I'm saying is, the dopamine neurons are involved in movements as long as the movements are strong, but not in fine-grained, well-controlled movements. So this is an issue. Are they involved in movements or not? It's very difficult to say, to answer the question without knowing what you mean by movement. If it's a controlled movement in the terms of precise motor control, no, they're not involved. If it's a movement like the animal moves around, like I move around with my hands, then of course they are involved. And we have labeled that motor activation. Or there's also sensory stimulation going on. There's a lot of stuff going on. That's very difficult to control. And that's what the neurons are involved. So the neurons have, in fact, multiple functions. Dopamine neurons do this reward prediction error to the reward and to the stimulus. And that increases with stronger reward. The dopamine neurons have then the additional attentional response. You can call it a salience response that got nothing to do with the reward itself. It's just the salience of the stimulus. It's just the impactfulness of the stimulus, the detection of the stimulus. And then there's the movement in blue, which can reflect a lot of stuff. It can reflect the movement, as I said. It can reflect the sensory stimulation that occurs, of course, during the movement, millions of receptors. It can reflect the back-propagating prediction error in temporal difference learning. We have seen a ramp like this with reward risk. It can be reward expectation, which can also be attention. It can be what we call parsimoniously arousal or behavioral activation. So the idea is then that the notion that one brain system has one function, visual cortex does vision, motor cortex does movement, that doesn't work with the dopamine system. The dopamine system has multiple functions. And if you want to see it from a constructive point of view, we can say that the multiple function of dopamine neurons is appropriate for a very old, evolutionary old system that still works even when we have new environmental demands because the organism has developed, it has evolved. Dopamine neurons exist in fruit flies, and they exist in all mammals, and they exist in all vertebrates. And of course, as you go up the evolutionary ladder, the demands on the neurons become more and more high, increase more and more, and the neurons seem to add these fancy functions like reward prediction error on top of these very primitive functions of movement or so. That's the idea. Okay. Now, I have a few more slides that relate to neuroeconomics, and that is justified by the second reward function, which is called approach behavior and choice. I don't know how long I should go on. It's much less than before. Maybe we have a few more minutes. Maybe I start and you can tell me in a moment whether I should stop. Okay?
That's okay, Professor. You can go as long as you want. That's okay. We have about 40 minutes left of the session.
Oh, I will surely not take 40 minutes.
Okay.
I'll be tired in
Okay.
40 minutes, as everybody else. Okay, let's get started to give you a feeling. Okay, here we go. Rewards are subjective. I mentioned that in the beginning, and I don't stop telling you that. You eat one steak, you eat a second steak if you want, but you surely stop at the third steak. Although physically the steaks are absolutely identical. That means your reward value does not entirely depend on what you eat, but also on your state. Reward value is subjective. The reward, and you may not eat steak. My wife doesn't eat steak. So for her, the steak has no subjective value. I like steak, but I'm not surely not 3 steaks. And for me, it has some subjective value. So the value is subjective in terms of my own body, but it's also subjective in relation to somebody else. That's the whole idea of economics and utility. So here we go. We can infer the reward, the subjective value from choices. We cannot measure the value unless you look at the neuron, but people have not done that until 50 years ago. You have to infer subjective value, or called utility, formally in economics, from choice behavior. You can measure choices by the frequency of choosing A over B. It's also called preference. So when you measure choices, you have to be precise, because otherwise you get a lot of salad. And what we have done is write down the conditions. The option set, that is the set of all choice options. So at the top you have 2 choice options, at the bottom you have 2 choice options. At the top it is large and small liquid drops, and at the bottom is banana and a drop of liquid. The option set includes all options. There's nothing hidden somewhere else. The options are mutually exclusive. You can only get one and not both. The options are distinct and well separated. That's important. You have to be able to separate them. Otherwise, you don't know what you're doing. They alternate pseudo-randomly to not produce a series of choices that then has a serial effect. They appear simultaneously, so you don't get distracted or you don't need have different memory demands. And the cost of the option should be constant. You shouldn't suddenly make the one movement more complicated than the other. The cost can be different within the options. That's part of the value, but it should stay constant. So everything should be well controlled, and the action should be distinct from the reward. Otherwise, we have again another confound, like we talked before about the movement and the reward with the initial dopamine results. So now we can estimate subjective value, and we say with equal choice, so-called choice indifference, both options have the same value. That's an important point to assess the reward value of an unknown stimulus, of an unknown reward, relative to the known reward. You titrate until you get choice indifference, and then the unknown reward has the same value as the known reward. Okay, right. So here we go. This is just a real wild example. You see at the left-hand side, the animal is choice indifferent between banana and a small drop of Schulz. And then the next one, the second from the left, you see that the animal prefers the larger drop to the smaller drop. And the third from the left, you see that the animal prefers multiple pieces of banana from a large drop. So we're increasing our preferences towards the right. And the fourth from the left, we see that the animal prefers a large drop of Schulz to the the multiple pieces of banana. So you have a whole hierarchy of preference, which is shown on the right-hand side on the top. The big drop has the highest preference, and the single piece of banana is lowest preference. And then when we take this color code underneath these rewards, we can see that the dopamine response at the bottom to the stimulus that predicts these fractal stimuli that predict these individual rewards increase in the same sequence, in the same rank order. The highest response is the yellow one, and that's the one with to the stimulus for the big drop of juice, and the lowest response is the dotted line, which is the response to the stimulus predicting the single piece of banana. Right? So the dopamine neurons seem to code the value in these choices. It's a very intuitive situation, totally uncontrolled. Okay, so we have to do something controlled. We go back to economic theory, hundreds of years, and look at Bernoulli, who said that the gain in money that you have when you already have a lot of money is much less with the same unit of money that you gain when you have very little money. The so-called gradually flattening utility function. Okay. The gain in utility is the gray vertical extent, the vertical extent of the gray box, and the physical value, and that decreases as you go towards the right, but the physical value is the horizontal extent of the gray boxes, and that's absolutely identical. So to do this, to estimate the value of these rewards, the subjective value, we can use utility theory and try to find a utility function for rewards. But first of all, we have to realize or make sure that the monkeys actually know what they're doing. And that can be done in a very simple manner. At the very left, you see the monkey facing a choice between a fixed low reward and a option that has one or the other reward, only one reward, but either a larger or a smaller reward. And the smaller reward is the same as the left fixed reward, right? So the right-hand option is a 50% top or bottom reward. The left-hand option is a fixed low reward. So obviously you would say you agree that the right-hand option on the left side is better than the left option, and the monkeys do that. Not 100%, but they have a stochastic choice preference for the larger option. And that is not risk, because then the next graph on the left part shows that the animal prefers a top reward as opposed to a option that has a top and a lower reward. All right? So the animal is not just risk-seeking, the animal actually knows what it's doing. And the dopamine neurons on the right-hand side show the same thing. This is a response to the red option, which is better because the lower reward in the red option is higher than the lower reward in the blue option. And the response to the red option is higher. The dopamine neurons know what is better. And that is not subjective or objective, it's just like that. Okay? Called first order stochastic dominance. If you Google it, you get a nice description. This is, yeah. Okay? And then we can construct a utility function. There is a so-called fractal procedure which uses the choice and difference point between a risky and a safe reward to construct a utility function. The experiment is shown at the top, and the stimulus, and then at the bottom we get the utility function. On the right-hand side, we see the fat red line, that is the utility function. And we see the black columns as the dopamine responses that increase gradually and then really sharply. They certainly don't increase in a linear fashion like the dotted red line, but they follow more, not precisely, but they follow more the red utility function. We did the same experiment with reward prediction error, which became much clearer, but it's very complicated to present. So I spare you this. I can only claim that what is written at the bottom, that the dopamine neurons do a prediction error in utility. We can call it utility prediction error, which is the current utility, subjective value minus the predicted expected value, or expected utility, sorry. Right? And then if that works, we can make an out of sample test, which is always useful. People like that. So we take on the left-hand side, a choice set in which both options are risky, but the right-hand option is more risky than the other one because it's more spread apart, right? And the animal likes the more spread apart one, the more risky one compared to the less risky one. Not 100%, but it likes it more. And the reason is, or the reason is difficult to say, but it correlates well with the shape of the utility function, which is in the center. The utility function is S-shaped, as I just showed, which means when you project these gambles on the x-axis, you go up and then you go over to the left on the y-axis, you compute the mean utility from the utility of the two outcomes. You realize that the expected utility for the riskier option is higher than the expected utility for the less risky option, which translated in the choice language means the animal should prefer the riskier option over the less risky option, which is in fact the case, which is shown to the left. So the dopamine neurons reflect that. They have a stronger response to the riskier red option on the right-hand side compared to the blue option. This is not a risk signal. This is a value signal that is boosted by risk. So the dopamine neurons are actually doing here, following the subjective value here, where the objective value, the mean between the two options is exactly the same. So this nails the point. Now this is one result, but we had plenty of other results that go all in the same direction. Dopamine neurons do subjective value in this situation in which we can identify the subjective value. Okay, so we go back to our rate code and we see what the dopamine response would do. And the idea is that the dopamine response updates the options in a winner-take-all choice mechanism. So I'll quickly tell you, or you probably know this. The winner-take-all choice mechanism has two inputs, the blue and the red one. They compete with each other. They have a mutual inhibition and a threshold mechanism, in which case only the stronger input will win. So if, let's say, the value of option B is higher than the value of option A, then the option B will win the output, and the animal will choose option B and not option A. Sorry, that should say at the top, chosen option A. Yeah. Okay. And the question then is, of course, what goes into this winner-take-all mechanism? And the answer is, it's the rate code. The rate code is higher for one option compared to the other option. So the rate code makes very much sense for these winner-take-all mechanisms. And the winner-take-all mechanism needs an input that has a metric where something is higher and something is lower, and the rate code provides exactly that metric. And then comes the text at the bottom of the graph, and that says, that answers a question that I'm being asked all the time. What goes into these values? Of course, it's the amount of probability, but that's still physical. Then there comes the subjective modifiers. That's the utility. The probability is subjectively weighted. The effort is subjectively weighted. There comes a reference point. Risk is nothing other than subjective. The delay is subjective, and satiety is subjective. And then comes the big deal. At the bottom is what I call the environmental influences. The value is also influenced and decides the choice by the personal history, conventions, compassion, cooperation, a lot of things, up to idiosyncrasy, prejudice, superstition, parochialism, nationalism, and all this bullshit that drives people nuts and makes them very emotional. All that affects the value of each choice option that you have, and according to the sum of these values, you can make a huge linear combination. And the sum of that's what the value of option A and the value of option B is at the end of the day. And that's where we make our decision. Okay, good. And that's my last slide. I just want to show you how this is the dopamine activation in the striatum of humans that reflects the input of dopamine neurons. The function fMRI and the PET scan are imaging the presynaptic activity in neurons, and the strongest input is from the dopamine neurons. So we see this from the top left. The money, of course, that produces a nice response in the ventral striatum. The reward prediction era, Reed Montague, who was one of my very esteemed collaborators, did that, couldn't wait to do that experiment 20 years ago. Dorsal striatum in this case. Beautiful faces as opposed to standard faces activated the caudate, the ventral caudate. At the bottom left, beautiful cars. This guy Spitzer had a contract from Mercedes-Benz to test the brain responses to the beauty of the Mercedes cars. So they could develop more beautiful cars that activate more the dopamine neurons. And that response that we see here is a subtraction between the picture to some beautiful sports cars minus a standard car. Then pleasant music, Sartora and Isabel Perez and a number of people at the Montreal Neurological Institute are testing now MRI, at the time PET scan, in response to music that is played to the people that are undergoing the test. Dean Mobbs had a humorous comic strips as opposed to informative drawings that activates the medial ventral caudate nucleus. The famous placebo effect, of course, that has been repeated numerous of times, including initially by Petrovic at the Karolinska in Stockholm. A romantic love activates the brain. In this case, people were shown pictures of their romantic partner, and the brain response to that were compared to brain responses to pictures of people with whom these test persons have been acquainted for half a year without having developed a romantic relationship. This again is PET scan. These are, as I mentioned, these are always the subtraction, perhaps response to one type of stimulus minus response to the other type of stimulus. At the bottom right is the most high complex reward, the social rewards. People that have violated social norms by, I think, taking everything on the ultimatum game or something, were punished, and then this punishment, apparently a reward, have activated the striatum. Although I have to say, the striatum is also activated by aversive stimuli that probably reflects input from the amygdala. I think I'm done here. Thank you very much for your attention. And if you have time, if you want to, I still have time to answer your questions.
Professor, for your wonderful talk. Now we open up for the question, and we have one student that raising her hand. Okay.
Well, hi, Professor Schultz. First, I would like to thank you for this informative lecture. I've got a question about the role of dopamine in attention. You mentioned that dopamine neurons also encode attention, and in the work, you showed that the dopamine neurons' response to attention and value of reward are nicely separated by just the time intervals they happen. Um, I would like to ask whether you have any ideas on how to just dissociate the representation of salience and value of reward in other areas of the brain, and specifically in other methods, for example, in neuroimaging pictures.
Yes. Well, as you saw, the interval can be longer if you have these complicated tasks in which the animal takes half a second to understand in which direction the random dots move, and only at that time, at that moment at which the animal has an idea where it goes, can it have a reward signal. And so that was a fortunate situation. That was by no means planned. That was just a chance result, in which we suddenly saw these two peaks. Well, we've seen these two peaks before, but much less separated. So what you need is a situation in which these two peaks, in which the attention and the reward value can be as separated in time as much as possible. Plus, you need a signal that's very fast. If you have a signal that goes up and goes down after 5 seconds, then you cannot distinguish 200 milliseconds. And this is the point with some of the modern optical recording methods in animals, that the time resolution is not good enough. Then you need to have these tricks in which what you did here, separate these two processes through the behavioral task. In human imaging, there's a group in Edinburgh that is finishing a paper in which they have separated that with a very high, I think a 7 Tesla magnet or something, but it was a lot of work. So in the normal case, you will not see the difference between the attentional and the reward response. But if you change the reward radically and leave the stimulus the same, or the same impact, then you can see suddenly that this initial peak still stays and the later part changes. So what we did then in this case, we measured the later part, or we measured the first part. And I have another example which we published, in which we could really see that the animal, when he learns new stimuli, first has a strong attentional response, and only later the reward response becomes more visible.
Wow, this is really nice structure, yeah.
Yeah, and that has, of course, led to many years of confusion where people just thought, on one hand they said, oh, dopamine is only attention, and the other side they said, no, dopamine is not attention. All this incentive salience thing is all wrong. Well, incentive salience doesn't work because it does not recognize the prediction error. But the idea that there's an incentive response, a salience response in dopamine neurons, of course, that is very obvious.
Yeah, really, thank you. Also, I've got another question, which is about the second part of your lecture. You showed beautifully that dopamine neurons reflect subjective values. Great. You also mentioned in the first part that dopamine neurons represent attention. I've just came across some literature showing that the interaction between attention and economic choice is a subject of ongoing investigation. It is really complicated whether attention can causally affect subjective values or not. So I would like to ask, have you considered this issue? Have you considered whether dopamine neurons can show salience of alternatives, and how attention possibly can affect subjective values in terms of increasing or decreasing them?
I don't know. I think in principle, let's say in the brain, attention modulates brain responses. So that's been known since Mickey Goldberg did his studies 40 years ago. So you pay attention, in particular when you have focused attention to a stimulus, the response to the same stimulus increases. I think that is very clear. Nobody has done that on the dopamine neurons. I mean, nobody has done what you're asking for, whether the value response increases when you pay attention to the reward as opposed to not paying attention. So you could do simply this experiment where the animal, I mean, you need a monkey for that. Mice and rats probably can't be trained. You need an animal that doesn't pay any attention, and you show the stimulus. And then you have an animal that pays attention to, let's say, an outside position where the stimulus is not shown. You show the stimulus, and then the animal pays attention to the stimulus, and then the stimulus comes up. And I would bet you that the response to the stimulus is increased when the animal pays specific attention on the spot of the stimulus. But that is the attentional modulation of a value, almost a sensory response. That is, in my opinion, separate from the initial attentional response. The initial attentional response that precedes the reward response is kind of an alerting signal. It tells the animal there's something going on, pay attention. And then later the animal realizes, oh, this is a reward, and now I can evaluate the reward and decide whether it wants to react or not. So that's a different thing, attentional modulation of an existing value response, or a simple, stupid, totally unselective attentional response that is also in time slightly different from the reward response. I once wrote that there is a benefit, an evolutionary benefit. That means you see something is happening, you can prepare a reaction because it might be a reward. And when you then later realize it's not a reward, you can cancel it. because the latency to this attentional response is so short that you have not started the movement yet. But of course, if you realize that this is a reward to which you are paying attention, then you have already prepared your brain for reacting, and you can react faster in a prepared situation as opposed to a totally surprising situation.
Yeah, great. Well, about the experiment, I've seen a paper on this written by McGinty in 2019, published in eNeuro, showing that they say, well, there is some causal effect of attention on subjective values, but not using this task structure. They just show that the duration of gaze can impact subjective values.
Yes.
And yes, they do not use dopamine neurons, but I mean orbitofrontal cortex neurons, I think, in monkeys.
I'm not aware of that paper. That you said is eNeuro 2019?
Yes.
Yes, I will have a look at the paper. I was always waiting for something like that. Yes, that makes perfect sense. You fixate the thing, and then let's say you have 3 dots, and each dot comes up and produce some value. But when you fixate one dot, you get the value from that dot. You fixate the other dot, you get the value from that dot. He has not done that, but I was waiting for I had written a grant already to do that experiment, but then I didn't have the time. That's a perfect experiment.
Yeah, and also I had seen some evidence in this regard in human behavior experiments, showing that gaze duration can shift choice preferences a bit. That was a paper I think, done by RML in the Rangel group. I think published in.
You say Rangel from Caltech? No.
By Carrie RML, Orly Bomo, and Antonio Rangel, published in 2008.
2008?
Yeah.
What was
yes.
it about?
It's about biasing simple choices by manipulating relative visual attention, which is the title. And they show that, well, if they force human subjects to look at an alternative for a longer time, they are more likely to choose that alternative. Something like this. These are all the things that make.
Antonio was involved initially in that experiment that you just mentioned. What's the author? Rich, right? Yes.
Yes.
Antonio actually was involved in that experiment initially, and he maybe even started that experiment. It's all together. Yes. Mm-hmm. I know the story a little bit. I just didn't know the paper. It's a very nice story.
Yeah, great. So that was what I thought about. For example, if attention is causally affecting subjective values, then how dopamine neurons can illustrate this?
I wouldn't say it affects subjective value. I would say it's more like you look at a stimulus and then you realize that you process that value of that stimulus. You look at another stimulus, you process the value of that stimulus, right? And whereas when you just look, you have an animal that looks around and you present a stimulus, you don't know whether the animal is looking at it or not. They probably do a saccade to that, but that's a much less controlled situation. Yes.
Yeah, you're right. But by the way, thanks for.
It does not. The boosting of the reward response by attention is a different story than detecting the reward value when you pay attention to that. That's a different story. Yes. They're closely
exactly.
And I can imagine both results. But it's a very nice. It's a very nice situation. It's worth doing an experiment.
Yeah. Thank you so much. So I need to, of course, think about that much more.
Rich eNeuro 2019.
Yes. So of course I can also, if you would like, I could send the link or the name of the paper email.
I'll find it. Thank you.
Yeah, great. Okay. Thanks a lot.
Say that again, please. How? Differ from the reward function, artificial reinforcement? Not as much as you may think. It depends what you do with the artificial reinforcement model. If you use a prediction error, then you have the dopamine neurons that do that in the brain. If you do reinforcement without prediction error, I don't know whether that is possible, then that's a different story. The people thought often that attention does all the learning, and that is pretty much discarded since Scholar Wagner. And the other thing was the Hull's drive reduction theory, in which you only go for reward when you have a drive, like you lack water, then you drink, and that has pretty much been discarded as well. I think the artificial reinforcement learning models, they're probably usually TD models, and that is pretty good. There are a few exceptions with the dopamine neurons, but that's pretty much the dopamine neuron. That's the point, that the dopamine neurons are actually what the artificial intelligence people finally found out, only for reinforcement learning. The deep learning is a different story.
Okay.
The deep neural networks is different, yes.
Okay, thank you, Professor. And can I ask some AI-related question?
Sure.
Okay. I was wondering, the current way that we approach AI is we make a big prediction machine with all the information that is available for humans, and we make this prediction machine, and we use reinforcement learning to explore a little bit more and find new things in a solution space. I was thinking, what is your take on that? How do you see the way that we see artificial intelligence right now? And do you think that reaching super intelligence is an engineering problem, or do you think there are some fundamental things from the way that you see you study intelligence systems from their neuroscience perspective? Is there something there that we still don't know, or is there something in biological organisms that show us something about intelligence that we didn't understand yet? My question is, do you think there are some things that we have to look for, or what do you think those the fundamental ideas are, like the reward signals that help us do reinforcement learning?
The big question. I mean, we can discuss for hours, but I think we may take reinforcement learning too simple. If you look directly at reinforcement learning, then you have the dopamine reward prediction error signal. But I think, I don't know about this movement signal. That movement signal is very mysterious. It is related to behavioral activation or something. I don't know. The neuroscientists get all excited because it's related to Parkinson's disease. But in conceptual terms, it's not very interesting. What's interesting, however, is that the reward signal, the reinforcement signal, the reward prediction error signal itself is not the only signal that I think plays a role in reinforcement learning. It's also this attentional signal. And I cannot find that paper anymore, but Rich Sutton, the first author in the Sutton and Barter temporal difference learning, he had once a paper, maybe 1996, in which he showed that the initial, what we call the attentional response, plays a role in temporal difference reinforcement learning. I don't know the details anymore, but I have written a review on this, what I call the two-component dopamine response or reward response in Nature Reviews Neuroscience 2016. And I have been very explicit in speculating that there is an advantage of having this attentional signal, which goes somewhat in the direction of the answer I just gave, that this is a preparatory signal that then allows the organism to acquire the reward, or you could say to notice the prediction error, and that may sharpen the prediction error. Because, you know, in evolution, you don't have a clean design. You have a design that works a bit, you add something that works, and something doesn't work, you forget it. And at the end, this is a messy system. That there's first a attention response and then a reward response. But in fact, it takes a while for the brain, when it sees a stimulus, to figure out first, to react at all. There's something, detecting the stimulus, then identifying the stimulus, large, big, whatever. Then the sensory properties, color, size, texture, whatever. And then maybe identified, do I know the stimulus or do I not know the stimulus? What is it? And then you come and say, oh yeah, is it actually good or not, right? Can I eat it? Can I drink it? Whatever. All that comes step by step by step by step. And in reinforcement learning, we just say reward value, go or not go for it, or increase or decrease your synaptic strength. And I think these preparatory things that are not just noise in the system, but they accelerate the whole process by already preparing the brain. And I wonder if, I mean, this is gaining speed. I don't know if speed is a problem in these computers, but I think the stepwise processing by evolution, by a biological brain that has evolved over evolution, there is something behind it. There's an advantage of that. It makes sense that you have a system that first does a gross processing and then a more and far fine-tuned processing. Maybe something like that could be done in artificial intelligence. I don't know enough about it, and I've programmed computers 50 years, but not that stuff.
Okay. So do you think this attention mechanism that we have, attention signal, could be something like that could help us solve the exploration problem in reinforcement learning?
Exactly. The attention may already filter whether it's useful to explore. By the attention mechanism will then lead to an identification mechanism in which you identify the thing, and then you can decide whether you want to explore it or not, right? So maybe that's the second step. So I have several diagrams. I had a big review in Physiological Reviews 2015, in which I have 4 or 5 steps. First, detection, and then identification of the object, and so 4 or 5 steps until it come at the end to value. And before you come to the end, you can already cut the process short at the beginning when you realize, oh, there's nothing interesting. Even without valuation, you can already say, well, this is something I identified, I know it's not interesting.
Yeah. There are very important people in our community that think the other half of the problem that we have to solve is the exploration in the reinforcement learning.
Oh, exploration is fantastic.
Yeah.
Exploration is wonderful.
And what do you know about this attention mechanism that we haven't applied in the exploration in the reinforcement learning? Is there something in the neuroscience literature that we can go on and find something interesting about that we can apply to try to theorize or maybe do some empirical work on the exploration?
What are you specifically looking for? Attention, exploration, what exactly?
What we know about the attention mechanism, attention signal in the brain that can help us solve the exploration problem in the reinforcement learning.
I don't know. People haven't done that. Exploration has become popular because of temporal difference learning and the absolute greedy mechanism and all that stuff. Before, the psychologists talked about exploration, but I don't know the details. The attention, on the other hand, in particular focus, cognitive attention, has been explored in the 1980s by a guy called Mickey Goldberg. He is a professor at Columbia University. Now he was at the NIH before. He was a colleague of Bob Ward's, who didn't work on attention. And he had several modulation of neural activity by focused attention or extrafoveal, foveal or extrafoveal attention and all that. That was not related to exploration. And so that would be a new avenue, which would indeed be interesting to combine the two, because obviously they're very related. If you don't pay attention, you won't explore. And if you pay attention and you don't explore, you're not going to win anything. If you don't explore, you don't win anything. You just do the same thing until it's over.
Right. Yeah, thank you, Professor. So I think we're out of time. Just for the final question, I want to ask, what do you recommend if you were in 2025 and just want to start your career? What do if you have these curiosities and you want to understand the intelligence, what path would you go? Do you go study neuroscience, artificial intelligence? I don't know, something fundamental like physics, mathematics, biology. How do you start your career if you start into 2025?
Well, I think physics with a good background of mathematics, I mean an intuitive knowledge of where mathematics is easy, but you do physics is a super good basis for natural sciences. Unless you get lost in details of mathematical proofs, so that's not important. But then of course it's different. Biology and artificial intelligence. Artificial intelligence is like a cognitive science, basically unconstrained. You can get lost and you do nothing, and everything gets very complicated. And as you probably saw in the talk, I'm trying to make things easy, because then what I say, when a monkey understands it, then I can also understand it. You know, make things simple, and then suddenly the thing come out yet you never think about. But when I'm in talks given by cognitive scientists, I get lost. After 10 minutes, I'm lost, because everything is so complicated, and one complicated term is explained by or defined by another complicated term. And when I ask, then I get another complicated answer, and I'm lost. If I cannot handle these things, other people can. Okay, fine. But if you go for physics, you don't do that. You want to know the basics. And physics is wonderful, where it applies simple mathematics to physics. Then, of course, you can get all complicated when you go into theoretical physics. And the same thing is economics, by the way. Economics is basically physics for behavior. They use mathematics to describe behavior. And then it gets also quite complicated. Not always necessary, but that's good. But now, artificial intelligence is pretty unconstrained. You can do anything and you can get lost. Biology is very constrained. On the other hand, there are millions of molecules, and the whole neuroscience goes more and more into molecules, because everybody needs another project and it takes another molecule, and everybody gets lost in the molecules. So you can get lost in both disciplines. Then I really don't know what I would recommend. I think if you're really very easy in mathematics, if you're like the best in the class, in the university class, then I think artificial intelligence is probably a good thing to do. If you're not, I'm not sure. I've seen people, mathematicians, pure mathematicians, that were the first in the class at Cambridge, and they kind of go in between biology and artificial intelligence, and they're not doing anything interesting. They're big guys, but it's not interesting what they do. So they get lost in the details. In biology, you need to have, if you have a good formalization that you get from physics and mathematics, it helps a lot, I can tell you. I don't have a physics background, but I did an undergraduate mathematics when I was a medical student, because medicine was so boring. And that helped a lot. And then in the last 20 years, I visited my colleagues at Caltech that are economists, like Colin Kamara, Antonio Rangel, and Charlie Plott, and a number of people. And that helped a lot. But then with my mathematical background, I could understand the stuff immediately, where other people need half a year. And that helped a lot. So any formalization is very helpful. And then it really doesn't matter whether you go to biology or to artificial intelligence. And of course, if you want to become rich, you go to artificial intelligence.
Okay. So your advice is go and understand the fundamentals, the basics.
I think that's the thing. Understand the fundamental. Don't make it too complicated. It distracts you from the fundamentals.
Right.
And yeah.
But
I think it's interesting between the two chairs, to be between artificial intelligence and biology, or for me, between economics and reinforcement learning and so on, neuroscience. It's really cool.
Yeah. Thank you. That's the same advice that Rich gave us at the start of the class. Rich Sutter. Go and study the fundamentals.
Cool. Yes.
Yeah.
And you need to have fun. Yeah.
Yeah.
Okay.
Thank you, Professor. It was an honor having you with us with this wonderful talk, and we very much thank you for that.
Sure. You're welcome. You're most welcome. Okay.
Thank you. So I think we can end the session. If you have any words, we can.
Okay. I'm fine. Thank you.
Okay.
Yes.
Okay. Thank you. So have a good rest of the day.
All the best. Yep. Bye-bye.